Welcome to Environmental Fridays. It is Personal, Season 2. You can learn more about our topics and speakers by joining our Environmental Fridays Facebook group. This series is brought to you by Best Early, the Benton Spirit Community Newspaper, Andrews University Community Engagement Council, GC Scored, Every Piece Matters. Introducing your host, Dr. Desmond Hartwell Murray, and this week's co-host, Abiadon Henry. This week on Environmental Fridays, we'll be hearing from John C. Weber.
All right, so we want to thank you all again. Welcome to Environmental Fridays. It is personal. This is our second season. We had a very successful first season. And this season, we are back with 17 presentations, 17 episodes. So I'm excited about this. And I hope you have an enjoyable time with us here today. First, I want to welcome our new people. I think Gloria's been on here before, but I think Leslie, have you been with us before? I don't hear her. Dixie, Megan, I think Chantel has been with us before. So welcome to all of you as well. Our co-host for today is Abiadon Henry. I hope I didn't mess up your first name. But he was born, Mr. Henry was born in Mason Hall, Tobago. He completed his secondary schooling at Bishop's High School, which is one of the top schools there in Tobago. And then furthered his studies at the University of Southern Caribbean. He is, in fact, as of this past December, graduate, BS Biology graduate, magna cum laude. And he is currently doing online studies in public health at the university in the UK. I forgot what he told me there, but he could fill that in for us. He's known, really interested in nature, in health, herbs, nutrition, healing. And in 2021, he started his natural care business by the name of Dawn Effect Health, Natural Health. He is also, after he finishes his master's, he is interested in doing his doctorate in natural medicine. So it's an honor to have you here with us, Mr. Henry. Soon to be Dr. Henry. Before you introduce our speaker, however, I usually give a little bit of background about what Environmental Fridays is all about and intended to do. The intention is for us to help mentor and guide the next generation and the general public in general about the environment, about environmental issues. And we try to make that connection because I think it's a fundamental fact that life and the environment coexist. They both need each other. There's that relationship between the environment and life. And so one of the things we seek to promote is our humility to the environment. We depend on it. It is personal. And of course, we should be deeply concerned and be stewards of our environment. And so some of the topics that we will cover and have been covered in this series so far are listed as shown here. And again, this is what we live in. It is our environment. It is very personal. And so Environmental Friday seeks to bring awareness and enlightenment on this area of concern for us, not just globally, but locally and personally. And we hope that this will inspire creativity, concern, taking proactive action in terms of caring for the environment. So at this time, Mr. Henry, you can go ahead and introduce our speaker today.
Hey, Des.
All right.
You got the wrong picture.
Is that the wrong picture?
Yeah, that's Peter Wampler, my colleague here at Grand Valley.
Okay, I'm so sorry.
That's okay.
I'm so sorry. Let me stop sharing my video as well. Go ahead, Abadan.
Right. So fantastic morning to each and every one of you, or should I say exciting morning to each and every one of you. My name is Abiadon Henry. So that's Abiadon Henry. And I am in fact quite excited and happy to be a part of this morning's session. To kick us off with our Environmental Fridays is Dr. John Weber. So who is Dr. John Weber? Dr. John Weber completed degrees at Moraine Valley Community College, where he did his associates, Southern Illinois University, where he did his bachelor's and master's, and Northwestern University, where he completed his PhD. He is currently a professor of geology at Grand Valley State University, that is in Allendale, Michigan, USA, where he teaches and does research with undergraduate geology majors. He also teaches geology field school nearly every summer in Red Lodge, Montana, USA, and in Baku, Azerbaijan. His research involves using GPS satellite data and structural geology, geochronology, and geomorphology techniques to study active and fossil deformation in Trinidad and Tobago, in Slovenia, and the Circum-Adriatic, and in Azerbaijan. He also studies meteorite impact craters and regularly consults for the petroleum and hydrology industries. John was elected a Geological Society of America Fellow in 2014. Ladies and gentlemen, I invite you to sit back, relax, and enjoy as we invite Dr. John Weber on the floor. Dr. John Weber, the floor is now yours.
Okay, thank you, Abiadon and Desmond. I appreciate the offer to share some of this research with folks. We're gonna sort of take a little journey back into Earth history and into biology history. And this is pretty new research that I'm getting involved with several geology colleagues and several biology colleagues. So I'm just gonna go ahead and share my screen and start sharing some slides with you. Please stop and ask questions if you have them along the way. I don't have so many slides that we have to rush through this. And I am gonna have to kind of jump back and forth between the PowerPoint and my web browser when I show some videos. So it's gonna get a little messy. The hot links didn't work the way I wanted them to. But let's go ahead and start with the PowerPoint slides. All right, so I like the title that Desmond helped me arrive at here. Basically, I'm going to be talking about the fusion between genetics or biology and geology. And this is work that I'm doing with a number of colleagues. So I'm a geologist by training. Jeanette Arkle is a geology recent PhD graduate who's now a starting professor at Augustana College. Nigel Noriega, Mike Jowers, and John Murphy are all biologists working in Trinidad and Tobago. And Jenny and I both work in Trinidad and Tobago as well. So if anyone needs to contact me for more information, I put some contact information on here. and I'll just move on to the next slide. So recently, I've started teaching a course on the Americas that combines geology and biology. And the premise of the course is that we start with the supercontinent of Pangaea, and then we break up Pangaea. We reconnect Pangaea with Columbian trade after we move people into the Americas. And I'm co-teaching that course with a biologist, and I got really interested in some of the biology stuff that we've been covering and sort of uncovered this new emerging field that I'm just learning about called geogenomics. And it works like this nice little cartoon on the right-hand side shows the connection between biology on the left-hand side and geology and environment on the right-hand side. So on the right-hand side, imagine something like a mountain range changing in elevation, right? And that geologic change is going to have effect on the biology, which is shown on the left-hand side. This is a family tree or a phylogeny. And every one of these red dots on here would be a divergence event or a speciation event where one ancestor splits into two new species. And the cool thing about phylogeny now with genetics is that we can get ages on these divergence events or speciation events. So we have something that's very comparable to the geological record. We can go back in time with the biology. So if we go from the biology to the geology, the new name given for this field of work is called geogenomics. If we go from the geology to the biology, we've been doing this for a long time, and that field is called biogeography, okay? And so let's look at the picture down in the lower left-hand corner. I'd like to bring out some of the major geological features in the Southern Caribbean and Northern South America. So of course, we've got one giant river called the Amazon and one big river called the Orinoco. These are in the top 5 rivers in the world in terms of the drainage area and the amount of sediment and water that they carry. So these are really big rivers. And there's some really interesting biology that's been noticed across these systems. Someone by the name of Wallace, who was a contemporary of Darwin's, noticed that the same species of monkey had different colored fur on either side of the Amazon River. So the formation of the Amazon, which is, something like in the Miocene, the Amazon wasn't there. So the formation of the Amazon has definitely affected the biology on either side of it, as has the Orinoco. The Andes, similarly, were not here in the Miocene. That's like, we're talking 30 million years ago or so. We didn't have any Andes Mountains until after 30 million years ago. We didn't have a connection between North and South America till the Panama Arc was emergent. And that happened as recently as maybe 5 to 10 million years ago. And our talk is going to focus on these northern mountains. In Trinidad called the Northern Range, and Venezuela called the Paria Peninsula, the Araya Peninsula. There's a coastal range of mountains that runs along the rim of northern South America. And that's kind of going to be the focus of our talk. And just to talk about some other connections between biology and geology in the Caribbean, was proposed by a Cuban scientist and an American scientist that there was a connection from South to North America before the Panama Arc, before the Great American Biotic Interchange occurred across something called the Aves Ridge, which is now sunken, and some of the Greater Antilles Islands. So these biogeographic or geogenomic connections have been known for quite some time. What's new is that now we have genetics and phylogeny to basically more highly constrain problems like the Great American Biotic Interchange and the connection between North and South America across Garlandia, what's called Garlandia. So geogenomics is pretty new. Just within the past decade or so, people have started to go in this direction from the biology to the geology. And in this talk, we're going to go in both directions. We're going to fuse biology and geology going in both directions. Okay. This is an excellent paper for anyone interested in this. I could highly recommend this paper. It's got some nice examples, talks about the Amazon example and the Andes example in a very speculative way. So we're going to first talk a little bit about the geology of this coastal mountain range. This is my specialty. This is work I've been doing for the past several decades. And to set the stage, we have to know something about the Caribbean plate and how it moves relative to its neighbors to the north and south. And it's basically sliding through a gap between the Americas. In this picture, I'm showing the Caribbean plate as the stationary plate and showing the Americas sliding past, moving to the west. Either reference frame works. Basically, the Caribbean plate is moving through the gap between the Americas, or the Americas are moving westward past the stationary Caribbean plate. And all of these vectors on the map are vectors that are derived from geological data, the black vectors from mid-ocean ridges, and the white vectors are vectors that we get by measuring directly with GPS the relative motion between Caribbean and South American plates. And the picture gets kind of busy toward the bottom because this is the region of study shown in the black box. But fundamentally, what we have is a big sliding boundary like the San Andreas Fault that forms the southern edge of the Caribbean plate today. And there are some funny little jogs in that sliding boundary, just like Death Valley in California. We have these places where we have the sliding boundary steps in such a way that it's basically making what we call a pull-apart basin. It's sort of an opening, a hole in the earth that's forming because of the geometry of the sliding boundary. So here we are, the sliding boundaries going right through the middle of the island of Trinidad. We'll talk a little bit about the Gulf of Paria pull-apart and its effect on the topography, biology, and geology. And I won't talk too much about Tobago, and I won't go much further past the Paria Peninsula in Venezuela. So we're going to focus sort of on this region here. Okay, so this is a picture that's showing the players. Caribbean plate is shown with kind of the pinkish purple colors. Okay, so today the northern range is on the Caribbean plate, as is Tobago. The South American plate is shown with these green colors. And we've got a continental part to the South American plate and an oceanic part. And the boundary between the two plates, so this green stuff here is kind of in transition between starting on the South American plate and being transferred to the Caribbean plate. Probably really should be shown with the pinkish purple colors because the Central Range Fault is today the plate boundary. And here's this pull-apart basin, this opening that we're making because the sliding boundary steps from the El Pilar Fault across the pull-apart into the Central Range Fault, okay? What's happening at depth, if we take a deep view, is that as the Caribbean plate slides past South America, actually the oceanic part of South America is being torn off of the continental part. And that tear fault system is something that we call a step fault. Okay, so the focus or the locus of tearing today is just to the east of the island of Trinidad. And that locus of tearing is preceded by sort of a bow wave of deformation, kind of like the bow wave that forms in front of a ship. So we can think of the Caribbean plate as being a ship that's moving through the gap between the Americas. And as it does so, in front of it, there's a big bow wave of deformation. So that bow wave has passed through Trinidad. Currently, it is visible in the Barbados accretionary prism. And the active tear is just east of Trinidad. And I'll show you what, in the next slide, what effect that has on the topography. So this is precedes the last slide in time a little bit. When the bow wave was present in Trinidad itself. So this is going to cause contractile deformation, folding and thrusting, thickening of the crust. That bow wave has now passed, and the tearing is going to make a deep cluster of earthquakes. And that deep cluster of earthquakes we can still see. It's called the Paria cluster of earthquakes. Many of the felt earthquakes in Trinidad are really deep, which is why they don't cause a lot of damage. And they're related to this tearing structure at depth, which we call a step fault. So the passage of this step fault is going to cause sort of a wave of uplift and subsidence to migrate through the plate boundary. And we can see that in the landscape elements in Trinidad, which is kind of a relatively new discovery. So that's some pretty heavy stuff. We're gonna kind of look at more geography here. So I think all of you are familiar with the geography of the Northern Range, right? We have some tall mountains, El Cerro and El Tucush, but notice what happens to the elevations in the range as we move toward the Gulf of Paria, pull apart. The elevations are very systematically diminishing, decreasing, right? So one of these valleys would be Maracas St. Joseph Valley, okay? And then finally, when we get very far to the west, those valleys are drowned by seawater because what we're looking at in islands like Shaka, Shikari, and Monos are sunken former mountains. These are just the tops of the mountains that are sticking out of the sea. The rest of the range is sunken below sea level. And notice the very symmetric pattern on the opposite side, on the Venezuelan side. We see the same sort of decrease in elevation as we move toward this hole that's being filled in with sediment called the pull-apart basin. All of this happens because the sliding fault makes a step. Are you guys able to see my cursor that I'm moving around?
Yes, yes.
All right, cool. So our story is going to focus on these coastal mountains. The rest of the talk will focus on the coastal mountains. This is kind of a busy slide, but we have some data of two different varieties that tell sort of two different stories, basically. That if we look at the million-year timescales, what we see is the western part of the Northern Range has been unburied or exhumed at the highest rates. So we have the deepest rocks exposed in the western part of the Northern Range, the most metamorphic rocks. And we get that story from what we call thermochronology. That gives us sort of million-year timescale history of the mountain range, and it's unburial. A stands for apatite, Z stands for zircon. Those are minerals that we find in the rocks. And FT stands for fission track, and HE stands for helium. And I'll come back to these in a few minutes, okay? We have a new technique that allows us to measure modern erosion rates. It's called terrestrial cosmogenic nuclides. The nuclide that we use is beryllium-10, and I'll explain how this works in just a minute. And this gives us erosion rates over timescales of thousands of years. And it gives the opposite result. So notice that we get the highest erosion rates in the eastern northern range today in the Holocene, or over the past several tens of thousands of years, okay? So we get a kind of a reversal, and we think that this reversal is due to this bow wave passing through Trinidad, the step fault passing through Trinidad. The early effects and the late effects of the passing of this bow wave and step tear through the island of Trinidad. The million-year timescale stuff, thermochronology, I wanted to give you an example of one of the techniques. We use 3 different ones. This is one of them. This is an apatite mineral. This is the same, chemically the same material that makes up our teeth and bones, except that this is inorganic apatite. It's a calcium phosphate with a trace amount of uranium in it. And the unstable uranium decays, and it leaves behind a decay product called fission tracks. And you can see them here. These are big damage zones. When the uranium decays, it splits into heavy particles, and those particles energetically sort of crash their way through the crystal lattice and leave behind these damage zones. So another daughter product is helium. Uranium and thorium and samarium decay, and one of the decay products, every time they decay, they release a couple of alpha particles or helium atoms. Those helium atoms can be trapped in the crystal too. So the way thermochronology works is that each of these minerals and each of these parent-daughter systems has got what we call a blocking temperature. And at temperatures greater than the blocking temperature, all of the daughter products diffuse out of the crystals. At temperatures less than the blocking temperatures, all of the daughter products remain. And so essentially when we measure the parent to daughter, we can measure the time at which the mineral grains pass through the blocking temperature. And these tell us very important information about how mountains form, how and when mountains form. So that's the whole kind of gist of thermochronology, is knowing which parent daughters to use and knowing the blocking temperatures of those parent-daughter pairs. And from that, you can get a history of unburial of the rocks that are now at the surface in these high elevation mountain ranges. So these are our results going from on the west side of the Latin curtain, as I call it, in Paria. These are data that were collected near the fishing village of Mukuru. You can see that over these million-year timescales that the rocks in the west of the northern range and even further west in Paria have come up to the surface relatively rapidly, okay? And they came up to the surface, let's say, between around 10 million years ago in Paria. In Trinidad, in Western Trinidad, the rocks came up to the surface relatively rapidly at about 7 or 8 million years ago. And finally, in the eastern northern range, where the bow wave has just passed through, the rocks have come up to the surface not so rapidly and more recently. So again, we think that the gray shaded area on each of these graphs, the region of rapid cooling, so we're plotting just temperature versus time, right, for 3 different regions, is showing the passage of the step fault or bow wave as it makes its way eastward through the Trinidad, Venezuela region. Okay, so this is a map that shows those modern erosion rates over timescales of thousands of years. And you can see the red colors indicate erosion rates on the order of 100 millimeters per 1,000 years. And then the lighter colors represent erosion rates that are basically 10 times smaller. So slow erosion in the west, in fact, sinking in the west, rising and rapid erosion in the east is our interpretation. Okay, you can see that these are nice maps. They're really important results, but they're also hard-gotten results, right? So we had to go to the field, and Jenny Arkle did most of this work. Go to the field and collect samples from every one of these stream catchments, and collect samples for the thermochronology from many mountain peaks, many coastal outcrops, road cuts, stream valleys. So this has been sort of 25 years of research here to get these results. The way this terrestrial cosmogenic nuclide or beryllium-10 method works is that we have a mineral called quartz that's very common in the northern range rocks, SiO2. And when the oxygen and SiO2 is bombarded with cosmic rays, and this only works in the upper couple meters of the Earth. They're shielding below 3 meters, and we have no effect from bombardment by cosmic rays. But the cosmic rays basically transform oxygen into a new daughter product that's called beryllium-10. So the more beryllium-10 we have in the near surface samples, the longer those samples have been sitting in the near surface. That's the basic parameter, okay? So what we can do is we can go to a stream valley. I'm not sure which one this is. Looks like we're in the western northern range or central northern range here. We can go to a stream valley. We can collect some sand, and basically the sand serves as a proxy sample of all of the rocks exposed in the whole drainage basin. And we can get an erosion rate for the whole drainage basin from a sample of sand collected at the mouth of a stream in the drainage basin. And again, we're gonna separate out the quartz. We're gonna then count the number of beryllium atoms, and there aren't very many. We have to go to an accelerating mass spectrometer to count those atoms. And then with some assumptions and some calculations, we can get the erosion rates like I showed you on the map. And again, erosion rates are 10 times higher in the eastern northern range than they are in the western northern range today. Finally, the last part of the geologic story is the story of these fossil beaches that have been lifted out of the sea. So we're at a place here near where the Tampere River empties into the Atlantic Ocean, near Kumana or Redhead Village. And this is what we call a marine terrace. This is an uplifted fossil beach. So if we can get an age for these sands and gravels, we can say something about uplift in the eastern northern range. And we can indeed get ages. I'll explain the technique that we use, but I'll just take you to that beach. So here we are at the beach that I showed you in the previous slide is shown here. And this terrace, which we call, let's see, which one is it here? It's the 71. I guess 71 is the average of these two numbers. So it's Redhead Terrace number 2. And we get a very precise age that sand is 71,000 years old, and it's 21 meters above sea level. And we see consistently along the East Coast that we have these uplifted fossil beaches. We have some beaches around Blanchisseuse as well that are giving us a little bit of trouble. Those are the ones shown in green here. But along the East Coast, we have a series of uplifted fossil beaches. And in order to get an uplift rate, we have to know where sea level was at the age of those beaches. So let's go to Redhead or Kumana 71,000 years ago, or 71,000 years ago. So this is the Redhead Terrace. Sea level was 60 meters lower than it is today at that time, okay? And the beach is 21 meters above sea level. So we do some algebra, and red hedge should be on this graph somewhere. We can calculate an uplift rate using those data. And this cartoon kind of summarizes all of that complex geologic and mathematical gymnastics that we have to do. Basically, we've got beaches that are way up in the air on the East Coast. We've got some beaches around Blanchisseuse that are lower. We get uplift rates for these, and then we cross an axis kind of in the middle of the northern range, and we start to see evidence for sinking. Okay, we can't measure the sinking because we don't have the geologic evidence like we do to measure the uplift. We have the uplifted beaches. The beaches would be sunken. I guess we could try to drill for them or something, but we don't have that evidence at surface. Okay, and this is the technique that we use to get the ages like the one at Kumana Village for the uplifted beach. Basically, when that sand is rolling around on the beach, the sun is bleaching electrons out of these traps. Okay, so as long as the sand is rolling around, the electrons get bleached out, and basically we would get a zero age for a sample like this. We bury the sample, and the little bit of radioactive material in the sample, and we measure how much of that there is going to basically put electrons back in those traps. And then we collect samples in the dark. We have to use tubes and pound them into the fossil beaches, collect the dark sample, bring it back to the laboratory, and then we measure basically how much stimulated luminescence we get from those trapped electrons. And that gives us an age for burial of the sediment. Okay, so whenever the sediment stops seeing sunlight, we start trapping those electrons, we can release them, and we can get the age of burial of the sediment. And this technique is called optically stimulated luminescence. Okay, so that's the geology part. I'll stop and take a breath and see if anyone has a question at this point. All right, if not, I'll move on to the biology and genetics part. And the first part of this talk, we're going to focus on the famous guppies that live in the streams of the northern range. This is maybe one of the most well-studied species on the planet. People from all over the world come to Trinidad to work on these things. They've been coming for decades. Most recently, the work that's been done has been this geogenetic stuff, not yet connected to the geology. So the first part of our new work is trying to connect the guppy genetics to the geology. And I'm gonna get out of my slideshow here. And I'm gonna go to my browser. I'm gonna show you a couple of video clips. Okay, so let me show you this video clip to start with. You all see the screen still?
Yes, still
Okay.
seeing it.
Okay.
How do animals adapt? Can they adapt within their lifetime, or does it take generations?
This has never really been done in any other organism before.
We have genetic data, we have morphological data, and we also have ecosystem data.
So why do we care about guppies in Trinidad and being able to do these experiments?
With that powerful of a data set, you can find out anything you really want to know about the fish.
How many individuals does it take to lead a group? In fish, we found that it took about 5 to 10% of individuals to lead a group. But then we were wondering whether this could be extended to other organisms as well, and whether similar principles apply to human groups. The tools for behavioral analysis that we developed, the guppy was essential for doing this because they provide such an excellent study system here in the northern mountain range.
There's a theory of aging that suggests that animals that experience high risks of mortality should age more quickly and die at an earlier age. I could test that prediction.
Nature and nurture happen at the same time.
Fish who are highly lateralized are much better at multitasking.
What's happening in the streams of Trinidad is in many ways unprecedented.
There's limited places where you can get this type of data, and this happens to be one of them.
These are complex concepts which are not accessible to most people, and they're things that should affect the decisions that you make.
I was looking for a way to make evolution come more alive for my students. The Trinidadian guppies help make that possible for us.
We try to apply swarm intelligence principles that we have derived from biological systems to company management.
Data affecting global markets is sourced by wildlife and the forests that secure fresh water supplies for this island.
What we want is a citizenry that understands these subtleties of science.
The northern range in Trinidad is one of the most fantastic places to live that I've ever seen.
So that's a little plug for SII and my colleague Nigel Noriega, who runs SII, as is this next one. I'll introduce you to Nigel in this next really short video.
I'm Nigel Noriega, a biologist working to reach people through film. I'll take you on a journey that starts on the islands of Trinidad and Tobago, where work with wild guppies is reshaping core concepts in biology. Trinidad ecosystems answer questions ranging from how brains differ between generations to how lifespan is determined. This is possible because neighboring streams with similar communities have been isolated from each other for thousands of years. In this natural laboratory, scientists generate some of the most rigorous data ever compiled for a wild vertebrate. Over 15 generations of populations where we know every individual, its parents, offspring, growth rate, and detailed knowledge of its environment have revealed new information about the speed and mechanics of evolution itself. We'll explore why work with guppies helps us reintroduce populations, manage global fisheries, and use animal group networks to decode collective intelligence principles that we apply to people and corporations. Our expedition to this web of science information is made possible by people in Trinidad and Tobago and provides examples of how much their local biodiversity matters to science, education, and economies across a world that is changing life on these islands. I'm sharing lessons from a small fish to bridge large gaps in generations and cultures between global and local communities.
Okay, we'll go back to the PowerPoint now.
That's amazing.
Yeah, you all should know Nigel. He's an amazing guy who does really beautiful work.
Yeah, I've tried to connect up with him. I'll do that again.
Yeah, okay. So I'm gonna try to reshare this file. Let me see, let me get the right tab here. Slideshow, play from current slide. Okay. All right, so you met Nigel, you know a little bit about SII now. One of the smartest biologists I've ever met, and a super nice guy, and super fun to work with. So I would encourage you guys to reach out to Nigel if any of this stuff sounds interesting, and look for the Home of the Guppy video to be completed relatively soon. So the name guppy, actually, the scientific name for this fish is called Pasilia reticulata. The name guppy, the common name, is a name that comes from one of the first naturalists, a Victorian naturalist who worked in Trinidad and the Caribbean at the turn of the century and did a lot of really important work on fossils and living species. And his name was Lechmere Guppy. And this is pretty fascinating to read the history of this naturalist, kind of a contemporary of Darwin and Wallace, and doing very nice work in Trinidad, particularly on the fossils. Okay, so now let's talk about some of the details of the guppy genetics, some of the genomics here. It turns out that the genetics of the guppies living in the western streams of the Northern Range are more like the guppies in Venezuela than they are the guppies living in the eastern streams of the Northern Range. So as you know, the Northern Range drains to the west in the Caroni River, and to the east in the Oropouche River. Those two drainages are separated by something that's called the Valencia High. And the genetics of these guppies are not alike, okay? These guys, the Western guys, are more like their neighbors in Venezuela than they are like their neighbors just across the Valencia High, okay? And this map kind of shows, this is a map that's made from genetics. It shows the genetic closeness. So the closer the lines and blobs are, the closer the populations are in terms of genetic populations. And the guppies and the short northern streams that drain the Northern Range, there's a separate story there, but I'm going to focus on the eastern and western populations. This is pretty well documented, a number of different studies. In fact, some groups are now calling this a different species or a subspecies of Pasilia reticulata than this one. So maybe these have already had a speciation event from a common ancestor. This is based on the mitochondrial DNA. So mitochondria are these organelles that live inside all complex beings inside of our cells. And they're probably bacteria that crawled inside of other creatures at some point. So these have their own DNA. And when we look at the DNA of the mitochondria in the Western versus Eastern guppies, we can see that they derived from a common ancestor around 500,000 to 600,000 years ago. This is assuming some mutation rates. And we can see that the guppies in the East are not only genetically different, but they seem to be genetically younger than the guppies in the West. So the fusion between the geology and the biology, I'm sorry, it's only in words. And I'll show you this in the next slide. So during glacial times, basically Trinidad is connected to the mainland. And we think that during one of those glacial low stands, when Trinidad was connected to the mainland, the rivers from Guyana sort of headwardly, they grew in an upstream direction, as rivers do, toward the river attractor, the high topography, the rising topography in the Eastern Northern Range. So we think that the rising of the Eastern Northern Range has sort of attracted big rivers from Guyana to erode headwardly like gullies do, and to basically bring new guppies to the eastern side of the Northern Range. And then hypothesis B is a little bit more complex. And I think you get the idea from hypothesis A. Okay, so this is a picture showing at 20 to 25,000 years ago during what we call the last glacial maximum, when there was a mile of ice sitting on top of Desmond and I here in Michigan. This was all dry land, and both Trinidad and Tobago were connected to mainland South America. Sea level was 120 meters lower than it is today. And that's why we have South American flora and South American fauna in Trinidad and Tobago, okay? So again, during one of these low stands, we think what happened, and we have evidence in the offshore that there are big buried river channels that we can see in the offshore, that some of those big rivers sort of eroded into the rising eastern northern range and brought some new guppies with them from Guyana, not from Venezuela. Okay, this is the last part of the story, and I'm getting close to quitting time.
Okay.
Desmond, just tell me, you know, when you'd like me to finish. I have a couple more slides. I can stop at any time.
Yeah, a couple more slides is fine.
Okay. So this is work that was published with the group of biologists I'm working with, particularly Mike Jowers and John Murphy. And we have some snakes on Tobago that we don't have in Trinidad. So this is the scientific name for this snake. We have genetics. We have the full family tree for this snake. And we can see that the snake in Tobago diverged from its common ancestor. So we have a different snake in Venezuela that's related to the snake in Tobago. Again, we don't have this one in Trinidad. That didn't happen during the last glacial maximum, but it probably happened during one of these older glacial maximums. And we don't know exactly why this guy chose to bypass Trinidad or whether it was there at some point, and we just don't find it today. But also at 150 and 350 and 650, sea level was roughly 120 meters lower than it is today. So we have multiple times for these divergence or speciation events to occur. And here the biology can inform us regarding, well, I guess biology and geology are both informing one another here. But the cool thing about these family trees is we can get the timing of these speciation events, and we can relate it to something that we know about the geology. So I think maybe that's a good place to stop. I won't go into those last two slides. That's a separate story that will take about 5 or 10 minutes. And I think this is a good place to stop.
Okay,
So,
very good, very good. Thank you so much. Obviously, we want to open up for questions. I have a ton. We got to bring you back. There is so much. Oh my goodness. I guess what I could say is, like, I've learned so much about my country that I didn't even know before. I knew about the existence, you know, and the connection with South America for sure. But the detail and stuff that you just outlaid there, oh my goodness, wow.
25 years of work.
Yes. Any questions?
Yes, amazing. I want to join with Dr. Marie to say that in our 16 years that we have spent here in Trinidad and Tobago, we have driven all over Cumana. I think we would have gone to those beaches. And just for you to talk about the high-rise beaches with the faucet, I need to go back and see that again.
Exactly.
But Doc, amazing. Thank you for this presentation, and very eye-opening and mind-boggling. But we reside currently in the Maracas Valley. So what implications does Maracas Valley have in this whole geological and the the scenario that you presented, what implication you did mention something in the beginning. Can you revisit and give us more
Right. So you,
on the Maracas Valley?
Yeah, Maracas St. Joseph Valley would be one of the
Yes.
western valleys.
Mm-hmm.
So it's on the sinking side of the northern range, and you would have this population of guppies that's more related to the guppies in Venezuela and older than the guppies living in the eastern streams, which drain into the Oropouche. So you're to the west of what we call the hinge line in the northern range.
And geologically, with regards to the faults and that you mentioned, what implications does that have? I know we are also in the earthquake zone very much. So,
right. So you would be feeling the deep earthquakes that I talked about in that deep tear. And by deep, I mean those earthquakes are a couple of 100 kilometers deep. So they're very deep. They can be pretty big, but they're very deep, which means they're not close to the surface. They don't cause a lot of shaking. Those are the earthquakes that you feel like a big thump, and then you don't feel much after that, right? And we've learned that the plate boundary, the central range fault, is what we call a creeping fault. So it's probably not able to generate big earthquakes. It's sliding day by day.
Wow.
That's the one that's south of you that I showed on some of the maps, right?
and the one that's going towards the east, towards the, there was one slide where I showed the sliding thing going towards the east.
Yeah, that would
up the Barbados and up that way.
That would be the central range fault. That's the big
the
sliding
central.
plate boundary.
Okay.
It's south of you. Goes right through the center of the island.
So just about a month ago, I was in Tobago and stuff, and one of the things we did was go up to the rainforest in Tobago, and we reached, I think they say it's close to 2,000 feet high above sea level, 1,800 or something. Okay. And when you mentioned that at the very beginning, that the geology affects the biology, you could definitely see that. Because as we went up and up, the plant life varied. And in particular, the ferns were up at the top, you know, like lots and lots of ferns and big, not little ferns, some huge ferns.
Fern trees.
Yes, exactly.
Yeah.
Exactly.
Well, that's the part of the talk that we didn't get to. There's a story that we're fleshing out. It's unpublished yet, about these tree frogs that live they only live above 700 meters elevation. And the sinking into the Gulf of Paria has separated a population of tree frogs in Trinidad from a population in Venezuela. We had a speciation event related to the geology that we just talked about.
Wow.
So that's another one where we're trying to get the family tree and to see how the timing compares from the geology and biology.
Dr. Murray?
Yes?
I would just like to know, in terms of the guppies that are here in Trinidad, how they are different from the guppies in other parts of the world or something like that.
I'm not the one to ask.
I think probably Nigel is the one to ask.
Nigel? Okay.
You saw a gentleman in the videos by the name of David Resnick.
Yes, I saw his name too.
He's really the guppy expert, the international guppy expert.
Really?
And he has published a paper recently in the annual reviews volumes about guppies, which is a fantastic read.
How is his last name spelled again? I missed that. R-E-Z?
N-I-C-K.
N-I-C-K, okay.
Yeah. I can send you the paper, I've got it.
Yeah, and resend me Nigel's info as well, because I did reach out to him when we were trying to put Environmental Fridays together, but I'll keep bugging him. Yes. Okay, other questions? We'll take maybe about 2 or 3 more questions? I have to get ready for another class.
All right. Let me start by saying also that this presentation has been very enlightening.
Yes.
I really did learn. I learned an abundance of information that I did not know before. And what I find quite interesting is that this fish, this little fish has so many different languages to evolution. And I think I heard that, you know, it has a link to, what is it, the climate as well, the environmental changes and so forth. So from the fishes, you can also talk about environmental change. Is that correct?
I'm not aware of that application. I'm more aware of the behavioral and sort of evolutionary work that David Resnick and his colleagues have done with the guppies.
Right.
So once he gives us the contact for David, you could reach out, I could reach out, you know, we could reach out to David to see.
That'd be awesome.
Yeah. Yep. Okay, anybody else has questions?
I have a quick comment here, Len. I apologize, my video is not working, so I can't show my face anymore. I don't know what's going on.
Okay.
I actually got bumped off earlier, and when I restarted, that's happened. So anyway, I in my former life, I did a applied botany degree at the University of the West Indies after I left Tobago before coming to the States. And one of the things that was emphasized that we learned in looking at the flora and fauna is the distinguished difference in the flora and fauna in Tobago than in Trinidad. And I didn't have all the geological information, but this clarifies it in terms of the connection between the two, between Trinidad and Tobago and the South American continent. Mm-hmm. So that's very interesting. And I didn't realize that even you mentioned the guppies from the east and west are actually significantly different because of this drainage and flow and range. That's interesting.
Mm-hmm. Mm-hmm. Yeah, when I go back to Trinidad, it's with a lot of different eyes. From this, you could, things that you took for granted, you see in a whole different light. Absolutely.
Well, okay. I'm glad people got something out of this. It's been super fun to learn and to sort of overstep my boundaries as a geologist and try to learn something about biology too. It's been really fun.
Oh yeah, that's where the fun is, right? Is when you step out of your own comfort zone.
Right.
And delve into, yeah. So the whole idea of geogenomics is really cool.
I like it, yeah.
I like it. All right, so we'll have to sign off, but we want to thank everyone who showed up for our first episode. It was a brilliant presentation, informative, interesting, engaging about things that we thought we knew. Trinidad, we don't, and there's more to come. Thank you so very much for opening our eyes to this. And we hope each one of you have a great weekend. We will definitely, after I work through things with Professor Weber, we'll be putting this up online on YouTube. Okay, thank you so much. Have a great weekend.
Thanks, nice to meet all of you.
Yes, thank you.
Thanks so much.
Yeah, bye.